Good morning, good afternoon, or good evening. I am Vinona, and this is my second ISA for biochemistry, highlighting the topic forces responsible for aDNA, bDNA, and zDNA conformations individually. Pictures of the double helix of the DNA have become so common that everyone is familiar with its overall shape and structure. This is called bDNA, and it represents an average conformation of DNA based on fiber diffraction studies. DNA is remarkably flexible as a molecule. Considerable rotation is possible due to the number of bonds in the sugar phosphate backbone and terminal fluctuation that can bring about bending, stretching, and unpairing of strands. Many significant deviations from Watson-Crick DNA structure is found on a cellular level, some of which may play important roles. DNA is associated with proteins, that is, histone and non-histone proteins, to form the chromatin. DNA as a whole is acidic, which is negatively charged, and it binds to the basic proteins, which are in turn positively charged, called histones. There are 3 into 10 raised to 9 nucleotide pairs in the human haploid genome, representing about 30,000 genes dispersed over 23 chromosomes for a haploid set. Let's now consider 3 families of DNA helixes. aDNA, which can readily form within certain stretches of purines. Example, GA, GG, GA, where G stands for guanine and A stands for adenosine. bDNA, which is favored by mixed sequences. zDNA, which is favored by alternating pyrimidine and purine steps. Example, CG, 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 where C stands for cytosine and G stands for guanine. The A and B DNA families are right-handed helixes, while the Z family has left-handed orientation of the helix. These different conformations of the DNA helix have very important biological functions. We can consider these conformations to be like a spring. Like a spring, it can be compressed and elongated. As mentioned, DNA is a flexible molecule, so it can be learned by its elastic property. aDNA. With reference to the spring, aDNA is a compressed spring. As mentioned earlier, it is a right-handed conformation. A-form DNA was first identified in the fiber diffraction studies of DNA at 75% relative humidity. In general, aDNA for any sequence is favored under dehydrating conditions, and certain purine stretches will favor A conformation even in case of higher hydration levels. A-form helixes are common for DNA-RNA hybrids. In addition, it is favored in triplex DNA. bDNA. Like A-form, it is a right-handed helix which was first identified in its fiber state at 92% relative humidity. It is famous as the Watson-Crick form, which you are very familiar with. Several sequences crystallized to high resolution have been found to adopt B conformation. zDNA. Unlike the A and B forms, Z conformation is found to be left-handed. They are not found in E. coli, but found in complex eukaryotes. The backbone is not a smooth helix, but it is irregular and zigzag in shape, hence the name Z form or zDNA. zDNA has been postulated to play a role as transcription enhancer and in terminal differentiation. In some eukaryotic genomes, 10% or more of the genome contains sequences capable of forming Z DNA. Thank you for watching.